Mobile DNA. Mobile DNA is the type of DNA that moves within the genomes. Most of the moderately repeated DNA sequences found throughout the higher eukaryotes. Most mobile DNA is in the form of transposable elements. Mobile DNA includes DNA-based transposons and retrotransposons. Some encode enzymes that catalyze the movement of the transposons. This is the different types of the mobile DNA. Mobile DNA can be in the form of DNA transposons. It can be in the form of viral-like retrotransposons, and it can be in the form of poly-A retrotransposons. So in case of DNA transposons, we see that there is a transposase sequence for the transposase. This is actually the element. And at the ends of the element, there are inverted repeats. Here is inverted repeat, and here is again inverted repeats. And in addition to inverted repeats, there is the sequence for the target site. In case of viral-like retrotransposons, there can be sequence for integrase and reverse transcription. There can be LTR, long terminal region. So this is the difference between the DNA transposons and viral-like retrotransposons, because here we see that there is sequence for the reverse transcriptase. The third type of the transposons is poly-A retrotransposons. Here we can see that is poly-A. So these are three main types of the transposons: DNA transposons, viral-like retrotransposons, and poly-A retrotransposons. If we see the detail of a typical mobile DNA or transposon, we see that there are two main components. In the middle, there is a gene for transposase, means that is an enzyme that produces an enzyme which recognizes the target sequence. And on both ends, there are inverted repeats. Here are inverted repeats, and here is again inverted repeats. So transposon include two components: gene for the transposase, and on both sides there are inverted repeats. And here are the target sequence. So this is the essential components of a transposon. Transposon are cut by transposase, that is an enzyme, and inserted on another site. Here is the example that this is the transposon. Here are the inverted repeats, and this region is cut with the help of enzyme transposase. And once it is cut from the original location on the genome, then this transposon that have a gene for transposase, that transposase identify the host region or the location where transposon have to integrate, and it cut that target sequence, and ultimately the transposon is inserted into that target region. The host fill the gap with complementary bases. So this was the transposon which was present in the original location, and this moves from one location to another location. Components of a complex transposon. In case of complex transposon, we see that there is a sequence for internal resolution site. There are gene for transposase enzyme. There is a sequence for a resolvase gene. So on both sides we see that there are inverted repeats. So this is the difference between the simple transposon and the complex transposon. The sequence for the transposase gene that produces the enzyme transposase that recognizes the inverted repeats and target sequence, while the sequence for resolvase gene produces the enzyme resolvase and it recognizes internal resolution site. Here is the internal resolution site. Mobile DNA also found in higher life form, like in case of maize, there are activator and dissociation elements. So activator elements and dissociation elements are found in maize. In case of activator elements, there is a sequence for transposase gene and there are inverted repeats. While in case of dissociation elements, there are inverted repeats on both sides. So in case of higher life form, mobile DNA is also present, and in case of the maize, we see that there are activator sequences and there are dissociation sequences, which are written as AC and DS. Movement of DS elements gives mottled corn. This is the example of movement of mobile DNA or the movement of dissociation elements. In the original color of the kernel, in case of corn, is purple. There is a gene for the purple color. While if there is a dissociation element, then the color of the kernel is white. While if the dissociation elements move from one location to another location, it gives the kernel color as mottled. So this is that how the mottled color maize looks. So this mottled colored maize is appeared due to the movement of the dissociation elements from one place to another place in the genome of the corn. Mobile DNA transposition. Transposition is the movement of mobile DNA. So this is a definition of the transposition, that is actually the movement of the mobile DNA that involves copying of mobile DNA fragment and insertion into another location of the genome. They are also called as selfish DNA. Mobile DNA in the form of retrotransposition, moving in the form of RNA elements or in the form of intermediate of RNA, coding for reverse transcription. In case of movement of retrotransposons or the retrotransposition, there is a movement through an RNA intermediate. So the examples are lines, signs, and retrovirus-like elements, long terminal repeats. This is an example of mobile DNA, which is also called as interspersed repeats in the human genome, where we can see that lines, signs, retrovirus-like elements, and DNA transposons. The percentage of lines, signs, retrovirus-like elements, and DNA transposons in the human genome. Mobile DNA is fuel for evolution. Probably the mobile DNA have significant effect on evolution by facilitating gene duplication, because in the previous lectures we have studied that there is a phenomena which is called as gene duplication, which provides fuel for evolution and exon shuffling, because as a result of transposition, where the DNA move from one location to another location of the genome, it is usually considered as that this acts as a fuel for the process of evolution.